Good morning, everybody. Let's settle down and get ready to start with the first morning session. It's a great pleasure to welcome you to the 6th Single-Cell Proteomics Conference. Can you click? And I like to think of this meeting as being a meeting place for different technologies. We think that there is a lot in common. The different approaches to studying the proteome can inform each other and help each other. And that's why today, today and tomorrow, you'll hear presentations that reflect technological developments in many different areas that are highly complementary. And they share some common challenges. And one of these challenges is the huge number of protein molecules present in a human fibroblast. So here in this tiny panel of corresponding to RNA, I have put in the ribosomal RNA and the tRNAs, and it's all piled together. And the protein bar is still standing tall. There are just way more protein molecules, and there is a much higher dynamical range of variation of abundances, which creates challenges. So let me illustrate this more pictorially. If you want to quantify a protein present in that fibroblast at about 8,000 copies per cell, just detecting one copy of that protein is equivalent to sampling this 3-liter bottle of 1 million yellow balls looking for one single black ball to find. And this is a difficult challenge for a relatively abundant protein. That challenge is faced by every single method, whether that's affinity reagents, whether that is mass spectrometry, whether it's some of the emerging, exciting single molecule methods. They all have to rise up to that challenge. And you may ask, how does it compare to detecting the corresponding messenger RNA for the same protein? It corresponds to about this example of 1 mil cube with maybe 10,000 yellow beads. You have to find one black bead. So in terms of sheer number of molecules to be counted, proteomics presents a challenge that is far more difficult than the challenge of quantifying the transcriptome. And those shared challenges have motivated this meeting now for a few years. Here I'm showing our first gathering in 2018. And it's so wonderful to see many old friends returning and many new faces joining us. Among the old friends, I want to mention Jeroen Kriksveld because of a very memorable conversation that we had in 2018. I remember after the meeting in the faculty club, we were talking about what we were doing back then, which seems very primitive compared to where we are now. But even back then, he was thinking very much about all the steps of sample preparation. Sure, you can miniaturize volume here, but what about the autosampler? What about this step? Aren't you going to have losses there? And it's just delightful to see his group and his students at this meeting presenting solutions to those very problems that he was articulating in 2018. So it's a very diverse community of many people returning year after year, new people joining us and providing new perspectives. And I like to think of this meeting as being highly synergistic, of being a venue for discussion where we can share ideas and hopefully we can borrow approaches that have worked very well in one field and then use the learned lessons to empower another field and uniting forces together. It's something like the Rosetta Stone, where we have to speak different languages. And that's a challenge. I'm not going to sugarcoat it. We are going to have presentations that might be a little bit too technical for everybody to follow. And I think we should put extra effort to make our presentations accessible. And those of you who may not follow everything and understand, please ask questions. Because our aim here is not just to share research. We want to do that, but frankly, you can do that by reading papers. What we want to do really is help build a community. What we want to do is to provide a forum for discussion. And that's why after every talk, there are at least 10 or 15 minutes for questions. And traditionally, that time is fully used up. So in our tradition, I'm sure it will be used up in this meeting. And we have many breaks in between the sessions where conversations continue. And I would like to encourage everybody to use those opportunities to engage and discuss and actively participate. I'll cover some questions. The first, some basic logistics for the meeting. The first one is very obvious. I already said we want questions. You're encouraged to ask questions at any one point. And in fact, we are going to have panel discussions for more questions based on a very positive experience last year of having a panel discussion. In addition to having in-person audience, we have very large online audience. And I would ask the online audience to mute their computers and not share their screen unless they're presenting. We believe in having well-caffeinated, well-fed groups. So there would be many breaks, and all the breaks are going to have plenty of coffee opportunities and snacks. And we've tried to have a wide diversity of different snacks, mostly geared. There is a bias towards healthy snacks, I'll confess that. But still a good diversity. We also have an espresso machine. If you happen to like espresso rather than Americano, you should try it out. We have posters upstairs where the meals are going to be, the meals during the day and the lunch. So I would suggest that those of you who would like to enjoy the posters, which I hope is most of you, take a look at the posters after lunch. Lunch begins at about noon, and we have until 1:30 to enjoy both food and look at the posters. And this year we're going to have a prize for best poster. It's provided by Parallel Squared Technology Institute, and Alex Petowski and Harrison Specht are going to be the judges of posters. So look out for them and do your best when they stop by your poster. Of course, I would like to thank our sponsors who make this meeting possible, and they make it possible for us to enjoy all the healthy snacks that they mentioned and all the dinners. We're going to a really good dinner place tonight. I hope you can join the dinner. And I want to again reiterate how important for me is to make everything in this meeting accessible. And to make it accessible, we are going to start with a very short overview of some of the technologies, especially newer technologies that have a higher energy threshold to be understood, such as single molecule methods. Nikolai Wanunu is going to give you a short introduction to them. We are going to talk about some of the exciting frontiers in those. And this part is really motivated of my experience at the lovely, beautiful Gordon conference, where talk after talk, everybody was sharing the latest cutting research that they were doing. But I was missing the basics for some of those. And it was really hard for me to appreciate all the new things that they have done. They have put one more brick on the top, and it's a beautiful brick. But if I don't know the base, it's hard for me to appreciate it. So the goal of this first session really is to give you a sense of what are we doing, how the technologies work, and make sure to ask questions if things are not clear. So I'm going to start with the first introduction and outlining some of the frontiers of what I think are frontiers for single-cell protein analysis by mass spectrometry. And I'll start by outlining a typical workflow. Again, that's a very short intro. I cannot include everything, but typically we start with isolated single cells. Most workflows start with those. We have to lysate and prepare samples, and there is an optional step of digesting or not digesting the proteins. So we're going to have some exciting talks this conference of people analyzing intact proteins. Then we can label them, which allows to multiplex multiple samples together. Then follows a very important step, which is the separation, normally done by nanoliquid chromatography or electrophoresis, and that's a very important step to achieve high performance, good results. After the separation, we have to convert the molecules into ions. And this happens normally by electrospray. There are other methods, and some participants are going to talk about those. And this is the Achilles heel, the least efficient step of mass spectrometry analysis. The step that mostly limits sensitivity is converting molecules into ions. Once we have ions inside of the instruments, these marvels of engineering produce beautiful data. They can detect down to single ions and measure them with very high accuracy and precision. And then, of course, we produce mass spectra, and from those we interpret protein abundances, sequences, modifications. And just to give you some very rough estimates of where we currently are with sample preparation, there is a wide range of different methods that have good representation at this meeting that can prepare anywhere from a dozen to thousands of single cells per day. And the analysis of those single cells is more limited in throughput at the moment. We can only analyze up to a few hundred single cells per day per instrument. And you're going to hear a lot of efforts and approaches of trying to increase throughput. That is something that is well appreciated as a frontier. And then in terms of how many proteins we can identify and quantify, the range is very broad, from a few dozen to many thousands. And part of the reason why this range is so broad is that it depends what we mean when we say proteins per single cell. Is it proteins detected? Is it proteins detected with high confidence? Is it proteins quantified with precision? Is it proteins quantified with high precision and high accuracy? And is it proteins that are supported by MS2 level spectra? Is it proteins that are not supported by MS2 level spectra? So there is a lot of variability in the reporting that explains some of that dynamic range. And there is another factor that explains this huge dynamic range, and that is that is the size of the cells, for which I'll have a bit more to say. But just to highlight some of the aspects where I think we can make significant improvements for all of these steps. For sample preparation, I think one of the big challenges is making it more accessible and more robust. While we have these methods that in principle are very, very powerful, they're very generalizable, everything is commercialized, you can purchase the equipment and repeat detailed protocols. That has not yet happened. So I think one of the exciting challenges is to really democratize and make this step much more accessible for everybody. On the side of protein analysis, I already mentioned the interest of increasing throughput, accuracy, and of course, detecting modifications in full-length proteins. A lot of the mass spectrometry analysis is done on digested proteins, and as Neil Kelleher is going to tell you, that's not the same thing as measuring the full-length protein. And in fact, he is going to tell us about some approaches to measuring intact proteins with spatial resolution. And on the side of data interpretation, I think that there is tremendous potential to make more of the mass spectra that we are interpreting. We have long made the point, and that point will be made again during this meeting, that only a small fraction of the detected mass spectra are currently being interpreted, so better algorithms could potentially substantially improve the depth of protein analysis. So speaking of that wide range of coverage of how many proteins we have per cell, this is a big part of the reason for it. Cells vary in size tremendously. And initially, when we started, we were here, we analyzed Jurkat cells in 2016, somewhere in the middle. And then we have moved both upwards to larger cells and downwards towards smaller cells. And some primary cells are substantially smaller, like orders of magnitude smaller than some of the more typically analyzed cells in the field. And this naturally makes a difference in protein content and the coverage that we can expect. So I'm moving here more towards the frontiers of what's new in the field and what are the things to watch for. One of those is analyzing more small and primary cells. Another one is increasing throughput, which can be done by a variety of approaches by using shorter separation. And we have cutting-edge experts representing the field of separations. You can talk to them to learn what is the newest and coolest research in that area. And then doing things in parallel, both analyzing multiple barcoded single cells at the same time and analyzing multiple fragmenting and analyzing multiple peptides at the same time. This parallelization allows to increase throughput. Analyzing post-translational modifications is a question that I very frequently get. Are we there? Can we analyze post-translational modifications? And the answer in some ways is we've been there from the very beginning, because if we take the very first data sets that we produced, shotgun data acquisition, we do variable search for post-translational modifications, and we can identify a few hundred phosphorylated, ubiquitinated peptides. And Ben Orsburn has also done this variable search with the data acquired on timsTOF, more MS2 spectra, a few more identified post-translationally modified peptides. There have been some approaches that we applied specifically to increase the detection of post-translationally modified peptides, such as the use of enriched isobaric carriers. In this case, we use enriched carrier phosphopeptides. And most recently, we've used prioritized analysis that, by using real-time control of data acquisition, can specifically sharpen the attention of the instrument to ions that correspond to post-translationally modified peptides, and that can increase the consistency and the probability of quantifying them. What I think is a very exciting frontier in that area is being able to analyze more of the modifications that we can biologically interpret, and being able to accurately quantify them and consistently across all of the single cells, so that we can support biological investigation rather than just demonstrate we can detect a few hundred post-translational modifications. And of course, the other big thing is, let's detect all of the post-translational modifications on the full-length protein. Again, that goes to intact protein analysis. Another frontier that, again, is well represented at this meeting is going to be spatial analysis. And some approaches for that have existed for a decade or so, but they have substantially evolved. Here, I've chosen to highlight work from Kristen and Ryan, where they've used laser capture microdissection to isolate small sections of tissue. This wasn't at single cell resolution, but obviously this approach can be further refined and pushed towards single cell resolution, work that Matthias Mann recently has picked up and worked on, and many other groups are moving in that direction. There are obvious challenges with laser capture microdissection that have existed for decades, and we have not yet solved those, but that remains an exciting approach to doing spatial analysis. Another approach is imaging. Imaging works very well for metabolites and lipids, as you're going to hear from Theo Alexandrov tomorrow morning. But some brave pioneers like Neil are doing imaging of proteoforms, and he's going to tell us about that later today. Again, that has not reached single cell resolution, but it's going that way. So all of these methods are moving in the direction of having higher resolution and higher depth of protein analysis. This is perhaps the most exciting frontier for me personally. I started my foray into this field with a lot of technology development that from the very beginning was driven by interest in biological questions. That was the motivation from day one. But initially, applying the technology to biological problems was quite challenging because the technology was just starting to crawl and walk. And now I think we have technologies that are much more ripe for biological applications. And as a testament of that, you're going to hear quite a few examples at this meeting of various groups and colleagues, and certainly from our group, heading in that direction and using the relatively mature, robust approaches, technologies to answer biological questions. I'm going to finish with an important pillar for those biological applications. And that pillar is putting the technology in the hands of all biologists who would like to use it. And that means making it accessible. This meeting early on was attended mostly from pioneers in method development, a lot of early adopters, but I think it would be wonderful to be able to open up the capabilities of what we have developed to a much broader biological community. And there are already important steps in that direction. but I think there is a lot more that we can do. One important step in that direction has been this community effort of formulating, articulating, and sharing initial recommendations and standards for protein analysis by single-cell mass spectrometry. And we've received a lot of positive feedback from the community for how useful those initial recommendations are. But we should keep in mind that this is just an initial step. We have much longer path to walk and go until we make the technology widely accessible to everybody who can benefit from using it. I'm going to stop here and invite Slavov Wanunu to give you a similar introduction and overview of what is exciting in single molecule protein analysis. So while Manny is wiring up with the microphone, I've just mentioned that he is a colleague here in the physics department and the bioengineering department of Northeastern, and the leader and pioneer in nanopore analysis for both nucleic acids and proteins. Earlier this year, he published a very exciting paper on being able to translocate whole intact protein molecules through nanopores. That's not yet sequencing, and Manny was the first to clarify that. There was a lot of excitement on social media. Finally, we can do intact single protein analysis with nanopores, and Manny was very moderated and very careful with his language to say, no, we're just translocating it through the nanopore. We're not yet sequencing it. But nonetheless, I think that's a very exciting thing that we can finally do this with proteins. And I look forward to hear from the expert what's happening in that field.
Thank you. If you don't mind switching over to the other keynote. So welcome, everyone. It's my pleasure to actually be here in attendance for the first time. I've been aware of this conference. It's happening here every year. And it's great to see a community that focuses on making technologies available for all and available, breaking the barriers of single cell proteomics. What I'm going to talk about is how various single molecule tools can be applied for studying proteins. There have been several breakthroughs or several sort of advances in the last few years. And perfect. Okay, great. And then the advancer. Okay, yeah, sorry. Not a multitasker. So, oops. Let's get through this. Okay. So as Nikolai has said time and time again, and this is a great thing to keep in mind, is that the number of protein molecules in a cell is huge. This number is just a kind of order of magnitude physics approach to thinking about these scales. And this is really a huge problem because when you talk about, okay, I have 1 billion of something or 100 million of something, the first kind of instinct is not, oh, let's analyze them one by one, right? Because that's the thing that you want to avoid in a way in order to achieve the throughput that you need. So the question is, how do we get information that's not available with other methods? And I think single molecule proteomics, which is basically focused on looking at proteins one at a time, but also looking at their, for example, full length information, looking at isoforms, post-translational modifications in proteins that are, I guess, at the beginning, more pure or more kind of, one class of proteins, for example. And the question is, how much can we get out of it? We won't cover the whole scope of everything, but I wanted to give you a snippet into what has been done and what has been proposed. So, for example, this particular review highlighted some approaches that are used for analyzing single molecule, for doing single molecule protein sequencing. You take your cells, your tissue, and you digest it, extract it, label it with some dyes, and then digest it or run it full length through a detector. And then these labeled sites would be detected, and based on that, you can do some computational analysis to recover the identity of where these labeling events occurred. So, for example, one can then take it down to a single molecule fluorescence tool and basically look at spatial information by either passing it through a FRET detector, for example. So, if there's for example, a donor on the motor that's moving the protein through the sensor, and then those dyes would have acceptor molecules. Then, when they reach close proximity to the donor, you would get a list of positions where those modifications are, and from that you could potentially identify many of the proteins. The second approach, which is the most kind of challenging one, I think, is nanopore sequencing, where you'd actually be reading the ion current through the pore. So this is kind of my technology, the thing that I've been working on for a while. It's basically, in this case, you're seeing a very primitive kind of a schematic of a pore, which is basically just a hole through a membrane. And you have ions everywhere. You have a pretty high concentration of ions everywhere. So when you apply a voltage, you get some electrochemical potential across the pore. The highest impedance is across the pore. So what you generate is a very localized electric field, which is achieved by ions that are basically traversing the pore. And when if molecules are able to linearize and go through the pore, one can recover information about one can say volume or the volume profile of the molecule as it goes through the pore. Another approach that has been worked on is tunneling. So this is electron tunneling. So passing electrons through the proteins as they are moving through a sensor, which is the tunneling junction. The advantage or the appeal of a tunneling junction is that you have a very high sensitivity to content between the two metallic contacts. However, one can imagine that passing a whole proteome through that would be extremely challenging, just as much as it would be through a nanopore. Oops. Okay. So I'm going to cover another recent technology that's, I think there's someone here from Quantum SI that's going to give a talk today or tomorrow. This technology is kind of exciting because it allows one to multiplex the detection of terminal amino acids that are on peptides. So if you have peptides that are bound to the surface of wells that are isolated, then one can use recognition proteins that bind and digest the terminal amino acid, but through the binding process and the binding dynamics, one can achieve identity of the amino acids. So that's a very exciting technology that we'll hear more about in this conference. Another approach is one can take a protein sample with the caveat that that protein sample is pure in a sense that it does not contain other proteins in it. And one can design nanopores. So I mentioned nanopores, and before what I showed you was a kind of a primitive structure, which is kind of a synthetic pore, if you will. But one can actually engineer protein nanopores to be very precise. And in fact, the original nanopore sensing was achieved on those protein pores, which are actually the sensors themselves. Here you're seeing a side view of a protein pore where some of the residues inside the pore have been modified in order to improve the sensitivity to different molecules. So in this case, different modifications that were hydrophobic achieved much higher resolution and much higher dwell times. And the dwell times basically is defined by how long a peptide resides in a pore. And by looking at the dwell times as a function of the amplitude of the events, the mean amplitude of the events, you can draw a scatter plot from this particular sample, which is lysosome, and you can see kind of a spectrum. And that spectrum corresponds to the different peptide fragments of the digested lysosome. And different variants of this protein pore have been used in order to improve the sensitivity and get basically, improved spectra. So this requires separations, right? Which is in itself a very challenging feat. But once you have achieved separation, then you can use this method already to identify peptides. Although, again, the caveat is that this has not been demonstrated for all the proteins, just a few. This is a very slow process. So this technology is moving much slower than the state of the art with mass spectrometry, but it's moving. no pun intended. And I'm going to show another example of moving a protein through a pore, in which case one can conjugate a peptide. This was published a couple of years ago, late 2021, where a peptide is linked to a DNA carrier, and the DNA itself can be moved by a helicase in a manner that basically provides a base-by-base ratcheting. So the enzyme is sitting on top of the pore, and it's bound to the DNA. It fixes the position of the DNA. It also allows the DNA to be pulled into the pore because the DNA is negatively charged. However, as you can see here, the peptide, which has to thread into the pore first, also has to be negatively charged or has to be introduced by some other means like electroosmotic forces. But in this particular example, this was a negatively charged peptide, which is another kind of limitation here. But what you can do then is basically pull the DNA molecule up using the helicase. And as it's being pulled up, it'll pull the peptide through the pore sensor. And this is a cross-sectional view of the pore where you can kind of see the sharpest region of the pore, which allows you to sense several amino acids at a time, maybe 6, maybe 8. It's not, the number is not exactly known yet. But each time this DNA motion occurs, it's discrete, and that's a real advantage. And that actually allows DNA and RNA sequencing using nanopores, because that discrete motion allows you to gather enough ion counts. And these ions are not the proteins; these ions are salt ions. You're basically accumulating salt ion motion through the pore as you're sampling a small part of a fragment of a strand. And traces like this are obtained where you can kind of see the DNA piece that's going through the pore, followed by the linker. And the linker molecule is just a click chemistry linker that links the DNA to the peptide. And then you can see the trace for the peptide. And basically, in the more recent demonstration, post-translation modifications were also detected. So you can detect phosphorylation on the same peptide and its position. However, the resolution is still not at amino acid level, and I'll mention some of these challenges in just a minute. I think we're up to the conclusion slide. So the current challenges, if we're talking about de novo sequencing with nanopores, single molecule level, I just wanted to say that we're nowhere near that. I don't think anyone is right now. It requires really resolving. I mean, this is kind of an overstatement maybe, but it's the safest statement to make at this point, but it requires resolutions that are basically, 20 to the 6th or 20 to the 8th power of current levels that you must distinguish. Of course, that's not entirely true because the motion, it really depends on how you move the protein through the pore. With DNA sequencing and RNA sequencing, it's the same problem, but it's been solved. The second point is that the controlled ratcheting through pores has only been demonstrated for a few peptides. So it's still not, even that isn't still not something that one can generalize and say that, okay, this is good for all proteomics. The third point is that when we talk about peptide identification, it does require protein separation. So if you throw in a bunch of proteins and you digest them and you just try to detect all of them and resolve all the peptide, you'll get a smear that you still won't be able to resolve. Many proteins. So we're still not at the level of being able to do what mass spectrometry obviously can do. But the point is that I think we should continue to develop these methods and see where we can go with there. Because honestly, a lot of us in the field did not think that DNA sequencing would be possible using nanopores. The last couple of points are one still needs to define a suitable pore. A suitable pore is one that allows you to discriminate and give you the best possible resolution. And that includes also the mechanism of motion of the protein through that pore. So it's not just the pore, but it's the whole system. And the last point, Nikolai really likes to continue to remind us of that, and that's a really important point, is the throughput. If you just, even if you have 1 million pores simultaneously working for 24 hours, you'll get 1 billion reads, and that's just about like all the proteins in a cell. Now, assuming that each read is not nonsense, and that's a very tall assumption, it's going to be very challenging. But I'm glad that we're working on it. and actually just today you got the notice of award on a new award to study that with to study protein sequencing with asymmetric pores. So we're looking forward to starting that. Thank you. Thank you.
Thank you very much for this introduction, Manny. Can you stay here a little bit more? I think we'll take some questions because, as I said, we love questions. And I have the first one for Manny, actually. If you can tell us a little bit about milestones that we should be watching for in the field of single molecule protein analysis. What are some of the big things that you think are going to be important steps towards achieving those goals here?
Yeah, I think the first milestone is really demonstrating robustly that one can detect single amino acid substitutions in a sequence, and I think demonstrating that for more than a handful of proteins. I think that's kind of something that if that's possible, then I think that kind of brings a lot of hope to the field. I think a more realistic milestone is, taking a class of proteins and resolving their post-translational modification landscape. So, taking some unmodified standards and making modifications, making a lot of standards. Basically, we need to calibrate our system for every possibility of molecular structures. I'll talk tomorrow about some of our research, and I think the milestones there. One of the milestones for me that's very dear to me is how to move the protein through the pore. What's the mechanism? So if there's a regular way to move a full-length protein through a pore, I think that really differentiates us a little bit from some of the mass spectrometry approaches, not all of them. But full-length protein analysis, I think, is a reachable or at least more appealing target than fragmented proteins, because you're actually then able to associate, as you said, modification landscapes and so on, and variants.
Thank you.
Yeah.
Are there questions? Neil, can somebody help with Georg, can you help with microphones?
So I have a lavalier, so I can.
Thanks, Nikolai and Mani. How might it change your future if you had a reference set of proteoforms to work off of?
It would be amazing. That's, most of our time is spent on making proteins, making different types of samples. One thing that I'll also mention tomorrow is modifying full-length proteins with terminal tags so we can draw them into the pores is also in itself a challenge. That's not so easy to do it's probably easier to do for peptides, definitely easier for peptides, but very much challenging for full-length proteins. So that's something we're also working on. In order to initiate the threading process of the protein, one has to derivatize the protein at the terminal end of it. And for full-length proteins, it's not as easy as at least. I would love to get your feedback more.
Yeah, let me just expand on that.
Yeah, sure.
A reference set would be something like some tens of millions of known human proteoforms. And in that set, could you infer more from your N-tagged proteoforms? Or if you knew a confined set, like my go-to is histone H4. Histone H4 human has 42 main proteoforms. And then if you get down on the iceberg, it has 180. But if you had that confined set, would that help the nanopore community?
I think it would. I think it would in the near future. I think even if I had that set today in my lab or in others' labs, I think that the challenge would still be how to drive them through the pore, how to move them through. So there's going to be some of these, if they were resolved, if they're all separated, for example, from that set. So if you'd have separate test tubes for each one of the forms, right? Is that what you mean? Or
the informatic as well. If you knew that they existed, your inference of this 6 to 8 amino acid probe region, I think you could infer more. That's my hope from that community.
Yeah. And
these two communities together provide immense synergy.
I totally agree with you. I think it would help. I think we're sort of getting the machinery ready for that. So I think it's going to be mature in a couple of years. I don't think it's mature today enough to take full length proteins and just be able to analyze all of them.
I think it's a good example of two technologies maturing together, right? We also don't have the 10 million proteoforms, but maybe by the time we have them, we'll have the nanopores working better, and you're going to cross paths to build something that's bigger, stronger, and better than either technology alone.
Yeah, it's in the works in several different approaches, but it's definitely not there yet, I would say.
Great. Thank you, man.
Okay.
Other questions? Have a quiet audience. Compared to mass spec, isn't this method rather cost effective?
Oh man, is anyone going to jump out at this? Well, I mean, apples to apples, if you wanted to compare throughput that mass spec gives you and throughput that nanopores give you, I would say that today, nanopores are not as cost effective. I think one has to really scale up and demonstrate a device that will do something at the same scale. And then I definitely think that it would be cost effective because, nanopores do not require expensive ion optics and, vacuums and so on. So yeah, I think it does have the potential to be cost effective, but it would be inaccurate to say it's cost effective today. Is my response.
I agree with that assessment. I think we are actually not ready to make it because mass spectrometry is usually used to analyze complex protein mixtures, and this is not something that anybody has ever done with the single molecule protein analysis method. So we don't really have a fair comparison that can be made. But in my view, they're going to be highly synergistic and very, very likely for certain applications. Perhaps very targeted, directed applications, single molecule methods can be cost effective and accessible, and that would be wonderful. I think that's the hope. That's why our community is pushing towards developing them. Mm-hmm. See a question at the back of the room.
So what do you think about the bottleneck of reaching the density of nanopore on sensor to be like 1 million pores? Or are there any ways to reach that? I guess from my impression, the Oxford nanopores flow cell have about a couple thousands of pores, and how to reach 1 million?
Yeah, I mean, there's anecdotes of achieving 1 million. I think one of the issues with nanopore sensing is that the bandwidth of what you're measuring exceeds by far the information content that you're actually measuring. So you're measuring many ions for each fragment, for each piece of protein that you're measuring, and that really is problematic in the sense that one has to then bus all of that data, right? So if you have 1 million pores, you'd have billions of data points per second, or tens of billions of data points per second. It's not clear yet what bandwidth of acquisition will be required. So I think the bandwidth is going to be an issue, but I think getting to 1 million is not impossible given the numbers of the data bandwidths of today. But I think developing such a device and implementing it requires a lot of effort and requires a good target application. And I think we'll probably see genome sequencing with 1 million pores before we see protein sequencing. So I think it's a good thing that genome sequencing is a target of nanopores, because I think it'll possibly match the requirements for protein sequencing.
Hi. I'm somewhat. All right.
I think you're close to a speaker.
So this may be a naive question. I don't know anything about this field, but I was wondering if people have considered using an in vitro translation system, basically using the ribosome itself to push a peptide as it's being synthesized through the translocon core as it actually happens in the ER membrane. Is that possible?
Yeah,
Can you just briefly repeat?
Yeah, sure. The idea is to basically take the in vitro translation using the ribosome to actually synthesize and feed the protein right through the pore. Yes, so that's actually the ribosome is such a complex machinery. It's going to be challenging to bring that whole machinery nearby the pore during the synthesis, and then make sure that the peptide gets in and not the mRNA or other kind of hanging pieces. But it would be amazing if one can do that. I think it's worth exploring that. I know that people are looking at ribosomes in other ways, like with waveguides and in single molecule context, and then using like labeled tRNAs to kind of see what tRNAs are incorporated. In terms of using that for direct feeding of proteins, I think it would be a great thing to try. I think it's going to be a little bit challenging to get the peptide into the pore in the context of the whole ribosomal machinery, because it's the nascent peptide will be there, and it'll have to be charged enough, or you'll have to have enough driving force to enter the pore first. I think that's going to be the biggest hurdle there, and once it does, if it's able to be pulled through, then in principle there's no reason not to do that. Yeah.
There's a really curiosity question. So, say for instance, you succeed in moving the protein through the pore. So, and this is based on, I understand, volume measurements or charge volume things. Would that be unique for each amino acid, and you would be able to distinguish without any bias?
Yeah, so if you're moving a protein strand through the pore, that's an excellent question, by the way. I'm glad you asked. If you're moving a strand through a pore, you'll never measure a single amino acid. What you'll measure is an assembly of several, or a strand, of several amino acids. And you don't necessarily move by one strand. You can move by 0.1 strands. So let's say you have 10 amino acids there, but you can move by one amino acid, for example. Then you'll be able to build up some redundancy and confidence as to what the context of or as to what the content of the next amino acid might be. That's what's done with genome sequencing today for nanopore sequencing. They're not reading a single base. However, I cannot answer whether it's possible or not, because no one has really demonstrated that. But yeah, it's a tough one. It's a challenging feat to achieve.
Yeah. Very nice talk. I just have a follow-up question. So, do you have any idea what is currently the best size of the peptide you are doing for this type of technique? Like, what is the size range?
So the different approaches that I mentioned, the peptide sizes range. It's for the trypsin digest. I think they're range from like 10 to 40 amino acid. But if you're actually trying to move the protein through the pore, what's limiting you actually, let me see if I can go back here or not. What's limiting you in this case is something very trivial that can be solved, but it's basically the spacing between where the motor, think of the DNA as a gear, right? If the DNA is a gear here, and this is your motor, your gear is going to end when it hits the end of the DNA. So what you'll be reading essentially is the distance from the pore, the peptide distance from the pore at best, right? To the place where the gear attaches to the motor. That is somewhere in the 30 to 40 amino acids right now, or maybe even less, 25 to 30 amino acids.
How about the lower end? What is the shortest peptide? Or you can just start with like a 1 or 2 amino acid?
Yeah, so there are also approaches which I have not mentioned. So you caught me on something I've not mentioned yet. But there are approaches where one can digest the peptide into amino acids and then look at just amino acid recognition. That's also a worthwhile approach, because like if you have a single amino acid, you're guaranteed to have 1 of 20 or whatever, the possibilities. And there, I think the challenges are going to be more of ordering, or ordering your sequence. So when you're trying to do like exonuclease sequencing, where you're digesting DNA base by base and then detecting each base, that has never actually been achieved. So it was, I don't know, it was discussed and maybe demonstrated somewhere, but not. It's hard to order your the sequence, of base by base or amino acid by amino acid. So I think that's the challenge. There is going to be localization in the form.
Thank you.
Good. I like to say the questions coming, but we are coming to the end of this first session.